Hello, everyone. Welcome to the first keynote session of UK Spine Conference 2021, our third annual conference. We're kicking off the first day, which is all about biomarkers, with strategic priorities in biomarkers for aging and multimorbidity. As I said, this is the first keynote session, and we've got two other great sessions this afternoon. I'm Leah Fitzsimmons. I'm the Patient and Public Involvement and Engagement Manager on UK Spine, based in the University of Birmingham. And I'm really pleased to see the broad audience that we've had sign up, including some of our PPI contributors who we've just had a fantastic session with this morning. So on to this afternoon, we're really lucky that we've got two brilliant keynote speakers who will be providing, I think, a nice contrast in perspectives on the rapidly developing area of biomarkers, what biomarkers mean, what they can do for aging research, and how we might harness their potential and create some powerful tools for the clinic. So just to note at this point, we'll be taking the questions to the speakers after both talks, but please do start asking your questions as they occur to you in the event chat, which is on the right-hand side of your screen. I might be the other way around, so sorry if I pointed in the wrong direction. I'll be picking up all the questions from there, and as I said, we'll put them to the speakers at the end. So up first this afternoon, we've got Thomas Jackson from the University of Birmingham, who is an academic geriatrician. His key interest is trying to understand the biological and clinical manifestations of successful aging versus what we might see as unsuccessful aging. So Thomas is going to give us his insight into how he started to use biomarkers and what it might eventually mean for patient care in the clinic. So Thomas, if you'd like to join us.
Good afternoon. Thanks for that introduction, Leah. So I've been asked to talk today, hopefully for the next 15 minutes or so, on emerging insights and imminent advances in biomarker research. Now, I'm going to take us on a bit of a journey to try and describe where we are now, so a broad overview of what we understand by biomarkers in aging research, and try and identify or highlight the areas where we need to go. And it's going to start off a little bit philosophically, really. So if we're thinking about aging, then we really need to try and define that and think about how we may describe that, or what our vision as researchers, as clinicians, as academics, as members of the public might think about aging. And this is a very crude graph. And you can see across this graph, this was the graph that I was trying to show you, and this concept that what you might call normal aging is associated with deficit with time. So we tend to lose things after we've gained them. And this concept that with healthy aging, what we want to do is push that curve out. So we want to push that curve right out to here. This is maximum health span. This is the dream. This is where we all want to be. And what I see in the clinic a lot is this curve dropping down. And this might result in a shorter lifespan, so somebody dies earlier, or they die at the same time, but they spend a lot of time in ill health. And pediatricians, so doctors that deal with children, look at this the other way, and they talk about this concept of failure to thrive. So they worry about children who are not developing, who are not accumulating things. And as a geriatrician, I worry about people losing things, or people declining quicker than they may otherwise do so. But that in itself is all very well. That describes a dynamic process. But if we're going to say that somebody is aging well, so this is healthy aging, or this is unhealthy aging, then we think about where those thresholds might be. So when do we begin to define somebody who is aging not successfully or in an unhealthy way? And a lot of that depends on where this threshold line is. So in other words, if somebody falls below this line of this skill, it becomes a problem. It gets in the way of their everyday life. And I've put this line quite high up here, and this is the line that it takes to do, or the ability it takes to learn how to do crosswords, for example. And you obviously don't know that when you're growing up, and you learn it as a skill. And then if we have this successful aging, so people that age very well, they will never lose that skill until they die. Whereas somebody that may be aging normally, say, or abnormally, they will lose that skill across time. So you might put your threshold for a complex task like driving, like doing a crossword. But for some people, that threshold may be lower. That threshold may be the ability to walk 100 meters, or the ability to be able to walk around their garden, or the ability to be able to wash themselves or get dressed for themselves. So that threshold, when they may then feel that they are becoming unhealthy, might be further down this functional line, and it might be across here. So again, it might take longer for somebody to drop that threshold here or here. But again, this person that's aging very healthily, then they never cross that threshold. So this is about how we define disease. And lots of things will influence that threshold. Some of that might be societal or person expectations. So if your expectation is that you are going to age healthily and you are never going to have problems where you may find things more difficult, you may walk slower, you may have more problems with your cognition, then the threshold that you begin to pick up these problems may be much higher. Or if we expect people to develop frailty, multimorbidity as they age, then that threshold becomes much lower if that's what we think is normal. Obviously, environment, socioeconomic effects can change those thresholds, as can beliefs about health, access to healthcare, and also diagnostic norms. And this really comes down to what is so exciting, interesting, challenging about doing any research with older people. And that is that there's no typical older person. As we age, we have much greater heterogeneity. When I'm teaching medical students, I say to them, if I measured something in you, like how quickly it takes you to run 100 meters, or how quickly it takes you to do a task using your brain, you will all pretty much do the same, and that will be boring to me. Whereas if I measure that task in 75-year-olds, there will be a huge difference in the person that doesn't do it so well compared to the people that do it very well. And this is what is the challenge as well of researching or understanding older people, because as people become more heterogeneous, it makes the science much more difficult. So what is unhealthy aging? How do we diagnose unhealthy aging? Well, we can talk about specific diseases. And there's a list, a couple of people have tried to make lists of what they consider to be age-related diseases. So diseases such as heart disease, type 2 diabetes, dementia. These are diseases which are much more common as we get older. And that's a disease in itself. But where do we cross that threshold with multimorbidity, for example? There's a number of different definitions of multimorbidity. That may be 2 diseases, it may be 4 diseases, it may be 2 diseases that affect our everyday functioning. As a geriatrician, we talk about frailty, and frailty is a syndrome where we recognize that people with frailty have a much greater adverse outcome to something that in somebody that was older but not frail wouldn't. So for example, if a person with frailty develops a pneumonia, they're much more likely to have severe disease and die as a result and not recover well, as opposed to an older person who's the same age but is more robust. We think about sarcopenia as a distinct disease of aging, muscle loss, loss of muscle strength, muscle mass, loss of muscle function as we age. And also, when do these problems become disability, so the inability to do something? So it gets really interesting. If we're going to describe healthy aging, we need to describe unhealthy aging. And as I've just been through, there's so many ways we could describe that. This Venn diagram here comes from a great piece of work, a systematic review, looking at the population of older people and looking at that population across a number of different studies to see how many of them have frailty, how many of them have multimorbidity, and where that overlap is. And the key thing is that if you take a population of people over 70, 50% of them at least will not have multimorbidity or not have frailty. So these people, in my mind, are probably aging successfully or in a healthy way. A group of people will have multimorbidity, so getting up to half of them, but only some of those people will have frailty. So when we think about frailty, this is not an inevitability. It's not what the older population looks like. So what does unhealthy aging look like? What does it look like clinically in a patient? Now, this is Daffy Duck when Daffy Duck was young and sprightly, and this is Daffy Duck at 78. And I think we'd all agree that you might describe Daffy Duck as looking frail here. He's unsteady, he's stooped over with a stick. And if we think about frailty as the number of people that have it in the community, actually in the community, in people over 65, it's somewhere between 10 and 15%. And even in people over 85, it's still only about a third. So again, frailty is something that I would consider to be unhealthy as opposed to healthy. But do we know what multimorbidity looks like? Difficult to know. Does Daffy Duck here, we've agreed he might look frail, but does he have multimorbidity? How can we see that? Does he have polypharmacy? And I don't think we've been able to describe what multimorbidity looks like as well as we have, for example, with frailty. So if we think about biomarkers, we need to think about what those biomarkers will do. Now, biomarkers can do a number of different things. They can diagnose disease. So if somebody has this biomarker, they have that disease. Biomarkers can also predict disease. So a biomarker could tell us that that individual was much more likely to get that disease compared to an individual who didn't have that biomarker. The other thing that biomarkers can do is they can tell us about the response to an intervention. So in other words, if I was to do a trial of physical activity, say in older people, I could find a biomarker that would reduce in people that did well from that exercise or stayed the same in those that didn't do well. The other use of biomarkers is that they can help us to begin to describe the biology. They can begin to help us understand what's going on in the underlying biology of people with these conditions. So a good example of biomarkers, not particularly in older people, but they may be some of the biomarkers that we use for cancer, for example. So PSA in prostate cancer is a good biomarker of progression of prostate cancer disease. But biomarkers don't exist in isolation, and we have to be careful what we do with them. We have to think about the ethics of them. Would I want to know as a young man that I was definitely going to develop Alzheimer's dementia, for example? I'm not sure I would, particularly if there's not a treatment. Would you, as an older person, want to know that your next 10, 15 years are going to be spent in ill health as opposed to being spent in health? It's difficult to know. So we need to look at some of the ethics around this. And biomarkers, we think of them traditionally as being blood tests, for example, but they can be any biological measure we choose, or they could be a physical measure or a cognitive measure. So there's a whole host of biomarkers out there. And you might think about physical biomarkers of frailty. So if we look at Daffy Duck, He's got a walking stick. That suggests he's got poor balance, poor postural strength. His knees are bent, so he may well have OA. Is he concealing a tremor of Parkinson's disease by having his hand in his pocket? He looks grumpy, which could be due to depression, pain, cognitive impairment. And he's got this stooped posture. So these are visible physical biomarkers of frailty. And interestingly, we know that if we measure grip strength, walking speed, or the time it takes you to get up and down out of a chair, these things are very good predictors of mortality. So these are good biomarkers of knowing whether we're going to die sooner or whether we're going to live longer. So these are biomarkers. This is a practical biomarker that we use in clinics to help us understand how people are in terms of their frailty. So people may be well or managing well with disease, or becoming slowed up, or becoming moderately or severely frail. In other words, people that need lots of help with their everyday lives. So this is a biomarker. And the other thing, and I haven't got time to touch on this, but I just wanted to mention, is if we're thinking about successful aging and how we measure that from a biomarker perspective, it's really important that we ask older adults themselves where that threshold is. What do older adults think is successful? What are their expectations? Do they like the term successful? Because kind of successful suggests that it's like passing or failing an exam, and it's something that you've got some sort of ownership over, but actually is it? So that's something to think about. So I just want to briefly touch now on the biomarkers that are out there and biomarkers that we can use in practice and particularly in research studies. So this is a proposed panel of biomarkers of healthy aging, which came out of an MRC project, and Professor Lord, who's within Spine, worked on this. And this was really a case of very clever people sitting around in a room looking at all the available evidence to see, well, which are good biomarkers that tell us whether people are going to age healthily. Now, I appreciate it's not come out very well on here, but some of this might be blood pressure. It might be how well you are doing blowing tests with the lungs, how good you are from a diabetic perspective, what your BMI is. It might be blood tests looking at your endocrine function, or memory or tests of your mind looking at memory processing speed, or these tests of inflammation. So this is a really good place to start for these biomarkers of healthy aging. So these are biomarkers designed to tell us whether we're aging healthily or not. Now, if we're going to talk about biomarkers of multimorbidity, then we run into a bit of a brick wall here. So there's been 2 recent systematic reviews looking at how can we predict or diagnose people with multimorbidity. And there's really been a paucity of research on this and limited research on these biomarkers. And really all these 2 could pick up with was obesity. And obesity may drive disease, or obesity may be a result of disease. You can see how that might be associated with having more than 2 diseases, for example. And then again, some of these inflammation markers, IL-6, CRP, and markers of endocrine dysfunction. But what I am really interested in is trying to understand the difference between the green person, the person that ages successfully with a good health span, and the difference between the red person on our graph, the person that ages unsuccessfully. And what we really need to be looking at, instead of saying a biomarker of age, because a biomarker of age is somebody's age essentially, but it's looking at the differences in these biomarkers between people who are frail and people who are not frail, or between people who are multimorbid and people who are not multimorbid. And this slide comes from a machine learning project which brought together 4 really big European, so 2 French and 2 Italian population-based cohort studies of older people, over 35,000 variable points. And this was cross-sectional, so this allowed us to diagnose, does this person have frailty or not, based on those blood tests. And they came up with 3. So proBNP, which is really a marker of cardiac function or stretch function. Cardiac troponin, which again is a bit of a marker of heart function. And then this soluble receptor for age. And this is looking much more actually at biological understanding of aging. So this begins to make sense having sRAGE here. What they comment here is that they said, look, we've got 35,000 data points here. We've controlled them using very clever statistics. We've got these really good cohorts, but the predictive power is undermined by participant heterogeneity. So what I said right at the beginning, what's exciting about aging makes aging research so challenging. So even with 35,000 data points, people were so different that to try and come up with really clear biomarkers of whether somebody is frail or not, was difficult. This is another, the BioSPHERE project, which is to look at people with sarcopenia and physical frailty. This again was cross-sectional. This was looking at proteomics. This was looking at cytokines. And 2 things came up. So 2 amino acids, tryptophan, and something that I can't read because it's very long. 2 cytokines, IL-1RA, which is actually an anti-inflammatory cytokine, and macrophage inflammatory protein, which is a cytokine of inflammation, so pro-inflammatory cytokine, and tells you about how activated your monocyte macrophages are. So again, this looks like this is quite good. We're beginning to pick some of these things out that can tell us the difference between sarcopenia and frailty. Now, what we've been able to do in Birmingham, and this is work from one of my excellent colleagues, another clinician scientist, Daisy Wilson, working with Liz Sapie and Professor Lord. And what she was able to do was, instead of just look at these soluble markers, these soluble messengers, was actually to say, well, what does this look like in aging mechanisms? So she looked at an aging mechanism and a cellular dysfunction that we see with age. And that's the ability of the neutrophil, which is the cell which responds to infections, how that travels to where the infection is. And we know that with age that goes down. And we know that you can rescue that by giving drugs affecting the PI3 kinase AKT pathway. But what Daisy was able to show is, if you take a group of older adults and you look at them as to whether they're frail or whether they're healthy, that some of these dysfunction is reduced with age, so with chronological age, but some of them are very specific to frailty. What she's shown here is that grip strength, so that marker of frailty, sarcopenia, of aging that we've shown, is associated with the ability of that neutrophil to get there, chemotaxis, as is a cognitive test, so a quite detailed cognitive test. So in other words, as your cognition reduces and as your grip strength reduces, the ability of your neutrophils to do their job also goes down. And that what she was also able to show was that when you rescue that with a drug, so with a PI3 kinase inhibitor to affect that pathway, that the rescuing of that was much more in the frail people compared to just the healthy older people, which again suggests that if we're going to look at some of these drugs and how we describe these aging mechanisms, we need to really know who's frail, who's unhealthy, as opposed to who's healthy. Now, if we go to a biomarker of prediction, so if I do a test, what will happen to these people in the future? And this again comes from that big Italian study, InChianti, and they looked at pathways. So taking lots of proteins, over 1,300 of proteins, they found 75 associations with the development of what they described as frailty, and they used loss of mobility. So over a 9-year follow-up, which people went from being able to walk 400 meters without it being a problem to not being able to walk 400 meters. And so that blood test at baseline, if you had dysfunction in your PI3 kinase AKT signaling, just as Daisy showed previously, that made it more likely that 9 years later you would not be able to walk 400 meters. alteration in your phagosome mobility, and alteration in that cytokine receptor interaction. So some of these are beginning to map onto the hallmarks of aging, these hallmark aging mechanisms. So this is to predict frailty. This is just to touch on a group of people working in geroscience trials. So this is looking originally at a trial to use metformin to treat underlying impairments in autophagy and nutrient sensing. This trial is just begun to start in America, the TAME trial. And what they identified was these are the blood-based biomarkers that we want to see a change in to show that metformin is working. And they've looked at markers of inflammation, markers of mitochondrial dysfunction, markers of nutrient sensing, kidney aging, cardiovascular health, so pro-BNPs come up. You've seen IL-6 and CRP in this talk already. HbA1c is a marker of metabolic aging. So these are the things that they think, although they haven't shown, but they would hope to show a difference in. And we've yet to see, because the trials haven't been done yet, what effect treating underlying aging mechanisms such as autophagy or such as impaired nutrient sensing, such as senolytics, what they might do to some of these biomarkers. And the last thing to be aware of is this platform based in the south of France, the InSpire platform, when they are looking to follow up older people around Toulouse for 10 years. Now they're looking at intrinsic capacity, which they describe as the opposite of frailty. So they're not interested in the negative, they're interested in the positive. They want to see who maintains their intrinsic capacity. So again, who has a healthy lifespan? And they're looking at markers of cellular senescence. They're taking blood, urine, saliva, pretty much everything they can get their hands on. So this is something to keep an eye on. This is the future. So if we go back finally to look at this graph again, this idea of what's unhealthy aging, where are these thresholds? This green person is what we want. This is where we want to get people to. We certainly want to get these people back to here, and then we want to get as many people into this area here. Now, what's the way forward? I think we need a clear vision, really, about what constitutes healthy aging and unhealthy aging. And I think geriatricians like myself have always said, well, we do unhealthy aging. But how can I understand unhealthy aging without understanding healthy aging? And I would put that challenge out to people who might describe themselves as researchers in healthy aging. But again, how can you describe healthy aging if you can't also describe unhealthy aging? So I think we've got to get together more. We've got to be able to describe this vision clearly. It's got to be transdisciplinary, and also older people's experiences and views of this has to be centered to this. Because as I've hopefully explained, we still don't really know how best to describe frailty from a biomarker perspective, multimorbidity from a biomarker perspective. We need to know what these people look like. I think the solution is unlikely to be simple. So even massive studies, however many thousands that they had there, clever machine learning techniques, they're not able to identify very well a simple 1 or 2 blood test marker. It's going to be more than that. And I suspect what we need to be focusing on is those underlying mechanisms. In other words, what's going on in the cell, how are the cells talking to each other? Because the readouts of those cell mechanisms, the soluble biomarkers, are probably not going to be helpful at this stage. So I think there's lots to do, but I think it's really exciting. And I think that as long as we've got patients, older people, and a whole gamut of transdisciplinary researchers, then we might begin to move forward. So thank you very much for inviting me. It's been really nice to try and put this together, and it's got a lot of my thoughts going actually. So I'm definitely looking forward to the talks. This is my research group, all academic geriatricians. There aren't many of us. We're trying to grow them in Birmingham, and Professor Lord, who's been an incredible mentor for me and her research group, and to UK Spine, who have given me some bridge funding to try and do exactly what I'm describing. So looking over time, in a well-described cohort of people who are healthy and unhealthy, what some of these biomarkers might be doing. So thank you.
Thanks, Thomas. That was really insightful. Thank you so much. So we'll move straight on in interest of time to Ivan Pavlov, who's joining us from the Medical Research Council, the MRC. And Ivan leads on initiatives to support multimorbidity research, and he's Secretary of the Population and Systems Medicine Board. And so we're going to get a completely different view now from the perspective of a funder, and as to how MRC are trying to develop and shape ageing biomarker research and the environment. So Ivan, if you'd like to join us.
Yes, thank you very much, Leah, for your kind introduction. I'll now try to share my screen.
Sorry, everyone. Just bear with us a moment. Thomas, are you still there on your mic or on your video? Would you take a quick question for us?
Of course, yes.
So we've had a question through from Raphael that frailty appears fairly late on in aging. So would it not be better to focus on biomarkers that tell us something earlier than much before somebody already becomes frail?
Yeah, no, that's a great question. And one of the things I didn't touch on exactly that was when your biomarker might be. You know, when would you do your biomarker? Now, the difficulty is that if you want to get biomarkers that are going to predict things over time, then you need an awful lot of time to do them. So, you know, the ideal would be to measure biomarkers in people as they age, and you might take midlife, for example, and follow them up. So it would be difficult to get really good results from that quickly. Of course, there's no reason why you couldn't do it, and why we shouldn't really be doing it, and we'd get a definitive answer further down the line. I think the point about frailty coming up later on in age, I think frailty is much more common later on in age, but we also know that younger people, there just is this phenomenon of working age frailty as well. And I think trying, in a sense, to understand people in middle life and who's going to get frailty or not is really important. If we look at the story of dementia and we look at the story of the amyloid hypothesis, it's likely that in a sense people have chased a pathological hypothesis, and they've been treating people who have got mild cognitive impairment, because in a sense you can't treat people that are asymptomatic because you can't treat everybody. But it may well be that one of the reasons why a lot of the drug trials focused on amyloid deposition are not working is because the disease has happened 20 years beforehand, and actually any treatment needs to be given much earlier. And I think it's likely, in my mind, that certainly preventative treatments for multimorbidity and frailty, because of course we can't prevent aging, are probably going to need to be given earlier on in life. But then, as exactly the question says, how do we identify these people? You know, because we can't give population level treatment, for example.
I'll follow up using Graham's question and potentially be a little bit provocative.
No, please do.
So presumably what we're looking for is some sort of sweet spot, so that we're not looking so early that we are creating ethical issues, like you say, of treating people who aren't sick. Where do you think the sweet spot might be? Would you go out on a limb and say, where do we want to be?
So going out on a limb, if we look at, for example, a lot of the immunological changes with aging, we look at the hallmarks of aging, you tend to be able to pick them up in people from sort of 50 onwards. If we look at development of multimorbidity, people tend to develop multimorbidity in that sort of, I'm going to call it late, middle, I'm going to call it early, I don't know, I don't want to offend anybody, but from kind of 50 onwards, So I suspect that that sweet spot is going to be around people in their 50s. And now, whether that means that you would then, to live successfully to 90, you'd need to be, say, having a treatment or an intervention for another 40 years, again, we don't know. But the ethical issues of saying that you're going to age unsuccessfully, but we can't do anything about it, I think that's the difficulty. If we can develop interventions that we know work, then it becomes easier from an ethical perspective to start looking for earlier biomarkers. But what they've done in dementia research is got stuck with having very good biomarkers in people that are healthy. But at the moment, we can't treat people, so it makes it very difficult.
So I think Ivan's just on his way back in. But
Great.
if you could give a few seconds comment on lifestyle and diet that Malcolm's asked about.
Brilliant. I didn't talk about lifestyle and diet. And of course, I think any intervention in trying to prevent frailty or multimorbidity or treat it is almost certainly going to be multifactorial. It's going to involve treatment of underlying biological mechanisms, but it's going to involve lifestyle, it's going to involve diet. We know that poor diet and poor lifestyle are associated with people developing those diseases of aging that I've talked about. So dementia, diabetes. So they're very likely to be associated with the ongoing development of frailty and multimorbidity. So that's why there's, at the end, it was that call for transdisciplinary research. But I don't think a food supplement is going to be the only answer. I think it's going to be more complex than that.
But you are starting to include those things in the omics data and the broader data set.
Yes, definitely. Yeah, and certainly in all of our research to date and our research going forward, that's key. If your cohort is well described and broadly described, then you can answer those questions. If your cohort just looks at diet but doesn't look at biology or anything underneath, then you don't get the answers you need.
And a very brief follow-up to that, are there data sets that include things like microbiome, vitamins, amino acid loading, that sort of thing, as well as just basic diet?
Yes, there are. So the one I would highlight really would be Twins UK. That's a very good data set with a lot of information on diet, dietary lifestyle. And the beauty of that Twins UK cohort is they're all beginning to get older. So we're beginning to understand that.
Okay, and I think we've got Ivan back with us. Thanks for bearing with us, everybody. Ivan.
Thank you. Yeah, I hope it works this time around. Right. Does it work like that?
Yes, perfect.
Right, excellent. Right, so what I'm going to do today is I'm going to give you a brief overview how MRC is supporting research into multiple long-term conditions or multimorbidity. Thomas did an absolutely fascinating talk highlighting some of the key problems within the area, highlighting heterogeneity, lack of proper definition of multimorbidity, and mentioned that scientists are really sort of hit the brick wall in trying to identify the biomarkers of multimorbidity. So understanding all these things, and of course the area is a fairly new research field in general. What I'm going to try to do is to talk you through the through our funders' perspective and how we sort of what's been our efforts in the past few years in trying to sort of stimulate this field and move the research forward. So multimorbidity presents a huge challenge for the healthcare system. Obviously, it's a huge burden for patients, and it is also extremely broad and challenging research area. With still very little understanding of how and why multiple conditions develop, how they group together, and what can we do to treat and prevent them. So multimorbidity is one of the 7 what we call health focus themes in the Medical Research Council, and it's mentioned it is a current MRC delivery plan which has been published in 2019. It's also part of a broader sort of overarching agenda set out in the UKRI's corporate plan that articulates the goals and ambitions of the organization for the financial year 2021. So we developed these priorities in discussions with wider research community, such as this one, the input of the industry and health and care sector, government departments, universities, and other charities. And one of the main vehicles that allowed us to take forward some of the activities within this field and within this sort of space was the UKRI's Strategic Priorities Fund, and I will talk about our SPF program in a little bit. So this slide quickly summarizes some of the key steps that we've taken in the past few years since the original priority setting activities, which was led by the Academy of Medical Sciences working group, set out in the publication of their report Multimorbidity, a priority for global health research. So shortly after the publication of this report, MRC have joined forces with the Academy, NIHR and Wellcome and held a 2-day workshop that further explored the enablers and barriers of the research in this field. And this was followed by several pump priming calls that we've launched in the relevant areas, including a call that focused on the immune-mediated inflammatory diseases, global multimorbidities, and also a joint call with NIHR that focused on understanding disease clustering and how disease comes together and pattern together in the patients. So having these funding competitions provided us with some very helpful insights into the area. Well, first of all, we were really pleased to see the very high interest from the academic community in this field. We received a very good and healthy number of proposals, high quality proposals in each of the calls, showing that there's quite a lot of researchers and research groups either already working in the field or ready to move into this new field of science. The research proposals also demonstrated that there might be some duplication of efforts due to fragmentation of research across the country, with researchers not being aware of each other and the interests of each other and their efforts. And the assessment process underscored a few important methodological issues, particularly around the statistical design of studies, difficulties to capturing longitudinal aspects of disease development and their trajectories, potential data source specific biases, and the need of a sufficient scale generally of the data source in order to address research questions in such a highly heterogeneous patients group. And also issues around limited availability of linked data that would also allow to interrogate broader determinants of multimorbidity that would also require multidisciplinary expertise coming into the field. So we felt that a scaled up approach would allow better overcome some of these issues. And this led us to a program, to an initiative that was supported by the UKRI Strategic Priorities Fund. So this initiative, jointly funded by the Department of Health and Social Care through NIHR, was developed also in collaboration with other UKRI councils, and in particular with ESRC and EPSRC. And the general objectives of these initiatives were to really build the capacity of the multimorbidity work by building a network of research collaboratives that would address different aspects of multimorbidity research and come together as a network, also allowing to share their ideas and knowledge and different approaches to tackle this different problem. So the focus of this initiative being to have a multidisciplinary team science addressing complex multimorbidities with multiple health conditions in the same patient, using rather holistic approach to understand the contribution of a wide range of factors, not only limited by the biological factors, but also by socioeconomic, behavioral, environmental, and other contributors that also impact on the human health. And we also wanted this initiative to build on the UK's strength in harnessing data from very good population resources and clinical cohorts available in the country, and also linking those resources with other data resources to get a breadth of data. So what I hope this slide demonstrates you is how we were able to expand the geography of research into multiple long-term conditions, providing good impetus to the field across the entire country. So on the left-hand side of this slide, you see the joint MRC NIHR pump priming call, which supported five research proposals across the country. And in comparison, in the middle of this slide is the schematic that shows the geographical spread of the research organizations involved in 11 developmental, what we called consolidator projects that were supported through the Strategic Priorities Fund call. And you see here, so the SPF program was a two-stage competition divided into two streams, and I'm very pleased to say that the first wave of this competition, the first two awards, have been just launched last month. One, led by Professor Arun Hingurani at UCL, focuses on uncovering common mechanisms that underlie diverse diseases by really sort of triangulating relationship between diseases, drugs, and their therapeutic targets, and using different large data resources, including electronic health records, genome-wide association studies, and randomized clinical trials. The second study supported through this initiative takes a different approach and looks at a very well-defined group of population, looking at comorbidities and multimorbidities in the hospital patients. And by doing so, we hope we'll lay the foundations for new approaches to inform the design of the future care of these patients. So the current SPF program is, the competition still continues, and we will know the Wave 2 awards next month. So we also, as a funder, we also continue to develop our vision and support of research in the field of multiple long-term conditions through an active dialogue with other research funders and medical charities, through being part of the cross-funder working group. And this group has developed a framework that provides a vision how we as funding community will drive forward the multimorbidity research agenda. So this framework is published on the Academy of Medical Sciences website, specifically dedicated, web page is specifically dedicated to multimorbidity research, and which we hope could act as a helpful resource for researchers in this field to know about new initiatives supported by different funders. So the framework I mentioned, the framework I mentioned has two pillars. The one pillar aims to drive advances in our understanding of multimorbidity and mostly focuses on research priorities in the field. And the second pillar tackles slightly sort of softer approaches and aims to foster a change in research culture to tackle multimorbidity, dealing with just softer cultural issues and problems related to generally to review and pursue research in such a multidisciplinary area of science. I should also emphasize that MRC also supports multimorbidity research through its researcher-led response mode, with applications being invited to submit to one of our research boards. and in particular to the Population and Systems Medicine Board, which highlighted multimorbidity as one of its opportunity areas. So we have 3 board submission deadlines per year and support applications with various formats: research grants, program grants, partnerships, and grants specifically aimed at new investigators. So the board opportunity area means that applications that meet its description would receive an uplift during ranking process within the group of grants of a similar quality. So, and I would invite you to visit our web pages for the description of multimorbidity opportunity area. But here I just wanted to highlight a few things that we feel are particularly important within this area of opportunity. And one thing here is that we welcome research on multimorbidity at all ages, not only limited to the very aged population, but across the entire lifespan. We also welcome and support research on multimorbidity that uses approaches that range from population and experimental medicine studies to characterization of different models, which is in particular could be helpful when we're talking about some of the biomarkers and understanding mechanistic process that happens at the cellular level. And Population and Systems Medicine Board particularly welcomes studies that focus on underpinning mechanistic research to generate evidence in support of interventions that could successfully target multiple long-term conditions. Unfortunately, at this very moment, I'm unable to give any specific indications of the future strategy. So I'm just referring this in this slide to our last corporate plan published on the UKRI website. But we're expecting that later this year, UKRI plans to develop and be published in a form of corporate strategy. And this will feature several themes where UKRI can work together to address societal changes, including health, aging, and well-being. And over the last year, the councils across UKRI have been working to identify themes and programs within this challenge, and this work is still continue. We are currently waiting for the funding allocations from BASE, and at this time, the time frame for any new program remains unfortunately uncertain. But once the final situation is clear, we hope to have more information to share with research community. I just want to finish that with a couple of web links that I hope you will find useful with the MRC multimorbidity web page that has the collection of all our activities over the past few years in this field, and a link to a cross-funder working group multimorbidity resource at the AMS website. And if you have any questions, do feel free to contact multimorbidity@mrc.ukri.org with any questions that you might have. Thank you, and I'll stop sharing.
Thanks so much, Ivan. That was really great. In the interest of time, we are going to bring the session to a close, but I know our audience will really appreciate just understanding the thinking of MRC and that there's so much careful thought and opportunities that hopefully we'll see in the future. So thanks everyone for attending and for bearing with us. We'll bring this session to an end there. Please carry on talking. You can contact all of our speakers directly through Hopin and can keep adding questions, and we'll pick those up by email or through the expo booths. We'll be giving a quick 5-minute break just for everybody. So we'll be starting the next session in about 5 minutes, which is slightly late. So we'll see you all then. Thank you ever so much.